So just to show you, here's the template is dark blue. Here's the new strand being replicated. Here the incoming DNT piece. Here's the enzyme here. It's going to ratchet along, stitching it together. You'll see it's going to show you primase and DNA polymerase, and the ligase is going to build the leading and lagging strands. Then we'll look at the picture. Maybe this will help you understand the pictures, I hope. All right, here we go. 3 minutes long.
Every cell in your body is produced. Before each cell divides, it must copy its genetic material in a process called DNA replication. Understanding of DNA replication comes largely from studies of E. coli, bacteria that are found by the billions in your large intestine. Let's take a look at how DNA replication occurs in an E. coli cell. As we zoom in, we see the DNA. At the origin of replication, the 2 strands of DNA separate, serving as templates for making new strands. The result is a replication bubble. The bubble grows in both directions, forming 2 replication forks. Let's zoom in on one of them. Many proteins work together at the replication fork. Only some are shown. Here, the DNA is unwound, and DNA polymerases, shown in orange, build new strands of DNA. Original parental DNA strands are shown in dark blue. Newly formed DNA strands are shown in light blue. Because strands in a DNA double helix run in opposite directions, the new strands must be made in different ways. One new strand, the leading strand, is built continuously. The other new strand, the lagging strand, is built in pieces.
There's the primer in red.
First, let's focus on the leading strand. DNA polymerase builds a new strand of DNA by adding DNA nucleotides one at a time. Each new nucleotide must pair up with its complementary nucleotide on the parental strand. Adding new nucleotides works the same way on both the leading and lagging strands. Each piece of the lagging strand begins with a short segment of RNA, shown in red.
There was primase that was left.
A clamp surrounds the RNA and attaches to DNA polymerase, which builds the rest of the new piece as DNA. When the piece is finished, it is released from DNA polymerase. How are pieces of the lagging strand joined together? A different DNA polymerase removes RNA and replaces it with DNA. However, it cannot finish connecting the pieces. An enzyme called DNA ligase joins the pieces together. Growth of the leading and lagging strands continues on both sides of the replication bubble until there are 2 identical DNA molecules. Although bacteria are very different from humans, the process of DNA replication in bacteria is similar to what happens in your own cells. DNA polymerase, which builds the rest of the new piece as DNA.
So here's the double helix, the parent. That's helicase right there. Attached to it, there's 2 DNA polymerases. There's in a sliding clamp. It's the lagging strand. See how it loops around back the other direction, so they actually replicate at least in the same direction, but one is going toward the fork, replicating toward the fork, the other one away from the fork. There's the primer, added RNA polymer, and then the 3' then is extended. It's going in that direction here. Okay. Let's show you that. All right. That's. I think that's. I'll post this movie on Canvas. I think it's pretty good. So with having seen that, let's look at these complicated slides now. So we can not be too blown out by the slides coming. And again, I decide to use the level of complexity that our book uses, so I'm not giving you more than what the book says a maybe biology student should know. Yeah?
So was that green form that was split, was that the helicase?
Yeah. It was splitting it apart, yeah. Okay. Now there's a series of 6 panels here that are going to track DNA as it's replicated. Okay? Here's the first 2 panels. These are in your textbook. So let's just point out. So here's our parental strand. The direction of replication is going in this direction. We're turning a DNA chromosome into 2 sister chromatids in the S phase or interphase. There's the topoisomerase that will clip the double strand so it can uncoil the tension generated. Here's the helicase that will unzip. Now this is a simplified version of what you saw. That was more of a more complex version, but this will kind of unravel us a bit. There's helicase that will tend to ratchet this apart. You can see, all right, and we see here are 2 RNA primers added by this primase enzyme. It can add a primer to the template without extending the 3' end of a pre-existing strand. That's what primase can do because it's RNA polymerase to that capacity, so this is a type of RNA polymerase. Once the primer is attached, here's the 3' end exposed, okay? Once the primers are attached, the DNA polymerase can attack into the primer in blue here. You can see here's the polymerase enzyme with its clamp, and begin to extend the 3' end. The leading strand grows toward the replication fork. The lagging strand grows outward because of the antiparallel nature of DNA. Does that make sense? Okay? Let's look at panels 3 and 4 now. This is now further along. The helicase continues to unravel the double helix. Topoisomerase is again nicking the backbone so it can release the tension. Okay? We see the leading strand continue. The leading strand only has to be primed once. Once it's primed, it will continue to grow toward the fork as the fork processes the entire molecule. Okay? But the lagging strand has to grow away from the fork. So it's primed, then grows, then as it's more primed, it grows into the previous one. See what I'm saying? Let me sketch this for you to show what I mean here. I'm just going to show one fork of the bubble, and I'll take the drawing with both later. Let's say here's our double helix. Unzip that far. Okay. All right. Okay. Now the primer has to be attached, complement anti-parallel to the template. So that in this case, this primer is in this direction. That's going to be the leading strand, right? And this primer has to be affixed right there. The lagging strand for that one. So this will be the lagging strand. This will be the leading strand. Okay? Then DNA polymerase can start to grow this in this direction. I'll show with dashed lines here. Okay? All right? Now let's unzip it further. So we're going to go a little later in time now. The molecule is unzipped this far now. Okay? Does that make sense? So it's further unzipped. Okay? Here's the initial primer here. And now it's grown all the way to the replication fork on the leading strand. Right? Primed only one time. The lagging strand was primed here at the same distance, right? It grew out to here. Then it's primed again. So it has to be repeatedly primed and then grows here. And when the DNA polymerase reaches that one, it falls off. And each of these, this is what we call an Okazaki fragment. We just continue it. So it will be primed multiple times. This is just primed once, all the way to the end. Right? that drawing make sense? And I'll show you that. We'll show panels 5 and 6 here in just a second. Here's panels 5 and 6 now. Again, they're just continuing on. The leading strain just continues to grow as the helix case continues to unravel the double helix toward the replication fork. There's the topoisomerase making the backbone to release the tension. Okay? As you see again, primed. As more is unraveled, we repeatedly prime it, and the new strain grows. The connections are sealed by the ligase. Okay? And then as it's unzipped even further, here we have unzipped it from here to here in panel 6. Again, we have to prime it again, and DNA polymerase will extend that until it reaches this end and falls off. And all these primer sequences are replaced by another DNA polymerase until we reach the end of the molecule.
That's DNA polymerase 1 that gets rid of the RNA.
DNA polymerase 3 is the one that extends the 3' and the primer does the bulk of the sequencing.
Okay.
DNA polymerase 1, actually,